Hello students, welcome back to Biology Tutorials by Dr. Pooja Yaddav. In today's session, under human excretion topic, we are talking about urea cycle. This topic is very important from exam point of view. Graduation syllabus में इसके ऊपर एक question आने के काफी strong chances हैं. And also NEET में भी urea cycle के ऊपर questions expected होते हैं. So let's see what is a urea cycle. It's basically a cyclic process that involves 5 enzymatically catalyzed steps, as we will see in the next slide, the entire cycle. इस cycle के जो first 2 steps हैं, ये operate होते हैं mitochondria में in the liver cells, and the remaining 3 steps they operate in the cytoplasm. Now, if we talk about the significance of the urea cycle is that जो हमारी बॉडी में प्रोटीन कैटाबोलिज्म के बाद जो एमोनिया गैस जनरेट होती है, ठीक है, दिस इज हाईली पॉइजनस गैस, सो इट हैज टू बी रिमूव फ्रॉम द बॉडी, राइट. बट मैमल्स में जो कि टेरेस्ट्रियल एडेप्टेशन शो करते हैं, जहां पे पानी की स्केर्सिटी है, इट्स नॉट अवेलेबल इन abundance as for spices. In that case, ammonia is being converted into non-toxic water-soluble form, which we call as urea. And then it is subsequently excreted by kidneys in the form of urine. ठीक है? So basically, जो toxic ammonia है, that is being converted into less toxic urea, the nitrogen waste, in terrestrial animals, right? So this is what is basically a urea cycle. This is also called as ornithine cycle, or it is called as Krebs and Henseleit cycle after the name of the discoverers. Now the cycle uses basically 3 intermediate forms, which are basically ornithine, citrulline and arginine, and converts waste nitrogen generated in the body into less toxic nitrogenous waste, that is urea. ये जो यूरिया साइकिल है, ये यूजुअली एक्टिव होती है जब हम एक काफी हाई प्रोटीन डाइट कंज्यूम करते हैं और वी आर इन द prolonged state of starvation. So now let's see what is this urea cycle. As I said, the first 2 steps of the cycle, it operates in the mitochondria of the liver cell. So mitochondria में, if you let me take the pointer please. Okay, so all right. So if you see here, this ammonia, it combines with carbon dioxide and to generate the first intermediate compound, the carbamoyl phosphate, and this reaction, it consumes 2 ATP molecules to produce 2 ADP molecules. Also, this reaction is catalyzed in the presence of magnesium ion as the cofactor, and the important enzyme here is carbamoyl phosphate synthase 1, जिसको हम CPS1 भी लिखते हैं. So the first compound that is being generated is carbamoyl phosphate. And आगे हम देखेंगे कि यही जो step है, ये CPS1 enzyme वाला, this is the key regulation step also of the entire cycle. Now this carbamoyl phosphate, it combines with ornithine और in the और इसमें वाटर का मॉलिक्यूल रिलीज आउट होता है. दिस रिएक्शन इज कैटलाइज्ड बाय ऑर्निथीन ट्रांसकार्बामाइलेज एंड द नेक्स्ट प्रोडक्ट citrulline is being generated. Now this citrulline shuttles from the mitochondria into the cytoplasm, where it combines with aspartic acid to generate arginosuccinate. This step is catalyzed by arginosuccinate synthase enzyme. Again, another molecule of ATP is consumed here in this step and AMP is generated. Magnesium again is used as the cofactor here. So if you see this cycle here, in totality, 3 molecules of ATP are being consumed in the first step and then subsequently in the third step, and the byproduct is 2 molecules of ATP and 1 molecule of AMP is produced. Now this arginosuccinate then splits into 2 important compounds. One is arginine and the another one is fumarate or fumaric acid, and the reaction is catalyzed by the enzyme arginosuccinate lyase, और इसको हम arginosuccinase भी लिखा होता है कई बुक्स में. Next, very important step, the arginine undergoes hydrolysis, means in the presence of water, arginine, it changes into ornithine. The enzyme involved here is arginase. And finally, in the last step, ornithine जो है यूरिया को प्रोड्यूस करता है ठीक है एंड it moves and a part of it moves again back into the mitochondria to repeat the cycle. Now this urea is being taken by the blood to the kidneys where it gets filtered and released from the body as urine. So this is the urea cycle. Now if we see the key aspects of urea cycle, so as I said, the first two reactions of the cycle, it occurs in the mitochondrial matrix of the hepatocytes, and the remaining three steps, they occur in the cytosol. The key enzyme and the regulatory step, or as we say, the rate limiting step, is the reaction catalyzed by carbamoyl phosphate synthetase 1. In short, we say it as CPS1, where ammonia, when it combines with carbon dioxide, it generates carbamoyl phosphate, right? And this is the enzyme. Now, this enzyme, this is a regulatory step because this enzyme is activated by the presence of NAG, that is N-acetyl glutamate, right? Now, how is N-acetyl glutamate produced? This in detail I'll be showing in the next cycle. But just to finish with the key aspects of urea cycle, if we see the energy cost involved, so the process it requires four high energy phosphate bonds, means a total of 3 ATP is being hydrolyzed to produce 2 ADP molecules and 1 AMP molecule, right? So nitrogen waste. So this urea cycle, it generally produces around 20 to 25 grams of urea daily, which is being excreted out. And the clinical relevance, which is of utmost importance here, is if somehow this urea cycle goes defective, so it will lead to hyperammonemia. There will be a greater amount of ammonia being accumulated in the body, and as I said, that ammonia being a very toxic poisonous gas, it will cause brain injury. It can lead to severe encephalopathy and can also prove to be fatal if not administered, right? So now coming to that step of the regulatory step, I said the rate limiting step is catalyzed by CPS1 enzyme, which is further regulated by N-acetyl glutamate. Now, how is this N-acetyl glutamate being produced in the body? So the glutamate, which is basically the non-essential amino acid, which our body manufactures from the proteins, this combines with acetyl-CoA, and this reaction is catalyzed by N-acetyl glutamate synthetase enzyme, and it yields this NAG as the byproduct. Now, in humans, if we see this reaction of synthesis of NAG, this is strongly activated by the presence of arginine. So if there is high level of arginine in the body, it will signal that there has been abundance of nitrogen, there is abundance of protein, right? Which will further promote this enzyme to produce N-acetyl glutamate. And once N-acetyl glutamate is produced, it will switch on the urea cycle. It will allow the fusion of ammonia with carbon dioxide, the very first step to generate carbamoyl synthetase. So it is basically, if I summarize it, it is basically the higher amount of nitrogen in the body, right, being generated in the form of ammonium ions or ammonia produced by decomposition of high protein diet. So that raises the level of arginine in the body. This arginine, it promotes the formation of NAG, and further NAG, it switches on, it regulates the CPS1 enzyme, which catalyzes the formation of carbamoyl phosphate, the first compound by the fusion of carbon dioxide and ammonia. And subsequently, the urea cycle, it starts operating. So I hope this is very clear to you all. I think this proves important. Thank you so much.